West China Hospital, Sichuan University. So today, Dr. Abel will speak on the mitochondrial dynamic and cardiometabolic diseases. Please join me in welcome Dr. Abel. Please.
All right. Thank you very much for this opportunity to share with you some of our work in the area of mitochondria and cardiometabolic disease. So I start with this slide that just simply shows that diabetes amplifies cardiovascular risk. On the left are data from type 1 diabetes. On the right are data from type 2 diabetes. This is data from Scandinavia. And you see that whether it's death from any cause, cardiovascular death, coronary disease, death, or hospitalization, patients with diabetes will have anywhere from a 2 to 4-fold increase in cardiovascular disease. Interestingly, you look over here on the type 2 diabetes, you see that over time, and this is over a 20-year period, that there has been a reduction in mortality from cardiovascular disease. But even with that, in the normal population in red, the diabetics continue to have this increase in risk despite, improvement in treatment and therapy. So this is a very complicated slide, and I don't want to try to take it all in because I'm going to break it into chunks during my talk. But what I want to convey here at a very high level is that diabetes is a systemic disorder. And the systemic changes in diabetes lead to very specific changes in the arterial wall and specific changes in the heart. So let's start with some of the systemic changes that occur in the context of diabetes. And let's start with, say, the expanding adipose tissue depot in type 2 diabetes, which as it becomes expanded and becomes insulin resistant, there's macrophage inflammation, there's increased secretion of inflammatory cytokines, increased or altered secretion of adipokines like adiponectin, increased leptin, which enter the circulation. The adipose tissue depots increases the release of free fatty acids as it becomes more insulin resistant. This goes to the liver that drives increased de novo lipogenesis and leads to the increased secretion of triglyceride-rich lipoprotein particles, remnant lipoproteins, and increased coagulation factors. So that's in the circulation. In addition to that, diabetes leads to activation of the immune system, particularly shown here in the bone marrow, that leads to increased release of inflammatory cells into the circulation. So when you think of what happens to the heart or the blood vessels in diabetes, it's not just simply the increased blood glucose. It's really this broad kind of systemic abnormal metabolic milieu in which the vessels and the heart maladapts. So for example, at the arterial wall, in the face of hyperglycemia and hyperinsulinemia in the context of type 2 diabetes, the classical starting point is endothelial dysfunction, which is essentially a decrease of nitric oxide. And as a result of that, this then primes the endothelium to be more inflamed. There's increased entrance of activated inflammatory cells because of insulin resistance, because of glucotoxicity, and of course, inflammation, as I mentioned before. There's macrophage activation. These are lipid-laden macrophages that ultimately form foam cells that form this lipid core. This is an atheroma. So when this then ruptures, there is in fact a thrombus that leads to like a myocardial infarction. Importantly, although I won't talk about this in this talk, we and others have also shown that mitochondrial dysfunction in platelets and increased glucose metabolism in platelets also occurs in diabetes that actually increases platelet activation to again drive abnormal thrombosis. Now, I'm going to talk mainly about heart failure in my talk today. And I want to make the point that diabetes is a heart failure amplifier. So this is a classic study from Scotland that looked across the age span of people without diabetes in the blue bar and people with type 1 diabetes in the green and type 2 diabetes in the red. Just looking at heart failure hospitalization. And what you can see is that heart failure is a disease of aging. So the longer you live, the more likely you're going to get heart failure. And you can see that but this inflection point in non-diabetics is occurring somewhere between 70 and 80 years of age. But if you're diabetic, this shifts to the left so that this inflection in heart failure morbidity actually increases by about 10 to 15 years. So what basically diabetes does is that diabetes increases the slope of the trajectory from preclinical heart failure to clinical heart failure. And this is summarized here in this cartoon. So the other point I want to make is that if you actually look at what drives either atherosclerotic heart disease, atherosclerotic disease or heart failure at a population level, there are actually quite important differences. So again, this is a large study that was done in Sweden, and they were looking again over this 20-year period at coronary disease, acute myocardial infarction, stroke, and heart failure. On the top, the red are people with diabetes. The bottom blue are people without diabetes. So what you can see, the good news is that over time, we have gotten better at reducing coronary disease and stroke in patients with and without diabetes, although, of course, the diabetics continue to have more than the non-diabetic individuals. However, if you look at heart failure, you see a very interesting pattern that there's a decline in the general population that's pretty steady, but in the diabetics, it's actually flattening out. And this is actually, I'll show you later, that this is actually being driven to a large extent by the increase in obesity. So the other thing that they did in this study was that they actually looked to actually ask the question, if you looked at myocardial infarction, coronary disease, stroke, or heart failure, what was the relationship between that and hemoglobin A1C? And so you can see here that as hemoglobin A1C increased, you can see heart failure increased, stroke increased, and MI increased, but not necessarily as steeply as heart failure. When they then looked at blood pressure, you can see that blood pressure was a driver of ischemic heart disease, both coronary disease, MI, and stroke. But interestingly, it was not a driver of heart failure in this population study. Um, that is systolic blood pressure. With regards to diastolic blood pressure, it really was driving stroke. And they also looked at renal function. You can see that as renal function declined, so this is the glomerular filtration rate got lower, there's an increased risk of MIs, but importantly as well, there's a significant increased risk in heart failure. They also looked at LDL cholesterol, and you can see that LDL cholesterol clearly drives ischemic heart disease, as shown here. But importantly, you can see that obesity or BMI had a huge effect on increasing the risk of heart failure in individuals with diabetes. And so when they examined then coronary disease, MI, stroke, or heart failure, and looked at what were the leading drivers, you can see that for coronary disease, it was the duration of the diabetes, LDL cholesterol, hemoglobin A1C, and diastolic blood pressure. For MI, the big driver, of course, was cholesterol and A1C. For stroke, A1C and blood pressure. But for heart failure, you can see the big driver by far was body mass index or obesity, followed by then A1C and then renal function. So the question then is, what happens at the level of the cardiomyocyte in this abnormal metabolic milieu that characterizes diabetes? So what this cartoon is attempting to show is that this is a coronary artery, this is a plaque, and this is a cardiomyocyte. And so in the cardiomyocyte, in the context of diabetes, there's increased uptake of lipids, increased uptake of glucose that leads to glycotoxicity and lipotoxicity. You can see some of the molecular mediators shown here, like ceramide, diacylglycerol, and triglycerides, or advanced glycation end products, hexosamines. And also hyperinsulinemia will also activate hypertrophic and pro-fibrotic signaling pathways in the heart as well. The three main targets of these abnormal changes inside the cardiomyocyte are the EC coupling machinery, the mitochondria that we're going to spend our time talking about. And of course, the nucleus through both epigenetic and direct transcriptional mechanisms, all of which really lead to maladaptation of the heart in the context of diabetes. So it's important to note that cardiac mitochondria are uniquely susceptible to dysfunction in the context of diabetes. Let me show you some data that we published quite a number of years ago in one animal model of many with diabetes. So the Akita mouse is a mouse model of type 1 diabetes. These animals essentially were diabetic for 4 weeks only. And when we look ultrastructurally at the mitochondria, you can see very clearly that there's really a striking difference in mitochondrial cristae compared to the wild type animals. You can see here, there's really a significant reduction in mitochondrial cristae density. And again, this is just with 4 weeks of diabetes. And yet, if you look in other organs like the liver or skeletal muscle or the brain, we didn't see these changes after that fairly short duration of time. And so there's some really cool studies that others have done in humans that I think have really brought this point home very, very carefully. And this is what I call the ultimate human experiment. So there are patients who get heart transplants every day. And so you take a healthy donor heart and you transplant it into somebody who has either diabetes or were pre-diabetic or get diabetes after the transplant. And one of the things that the transplant cardiologists do is that they do cardiac biopsies at 1 month, 3 months, a year, just to see if there's any evidence of rejection. So a couple of groups in Europe took some of this biopsy material and actually looked very specifically at mitochondrial function. So this was published out of Germany back in 2021. And they looked at those post-transplant patients who were non-diabetic or diabetic. And so you can see they saw that the diabetics, and this is within about 3 months of transplant, had more diastolic dysfunction, had more myocardial edema. Panels E, F, and G and H are showing mitochondrial respiratory capacity. You can see all of that was reduced in the diabetics relative to the non-diabetics. The diabetics are in red, the non-diabetics are in blue. They also show that the mitochondria were uncoupled, and also show that there was increased expression of inflammatory genes like NF-kappa B and increased lipid peroxidation. So this is telling you that pretty early on, if you take a healthy heart and you put into a human with diabetes, you begin to see pretty compelling signals of mitochondrial dysfunction, mitochondrial uncoupling, inflammation that tracks with the onset of diastolic dysfunction in these hearts. The same group also looked at individuals who had pre-diabetes. So hemoglobin A1C is between 5.7 and 7, so not quite meeting the clinical criteria for diabetes. And what they also showed was that, and this is probably shown here best in panel E, that even in the recipients of a healthy heart who had pre-diabetes, again, you could see that there was already evidence pretty early on within 3 to 6 months of impaired mitochondrial respiratory capacity. Another group did a much more comprehensive study. This was the first one actually of this sort. And what they looked at was just the accumulation of lipid in the hearts of individuals with transplantation. So they had the recipients who did not get diabetes, recipients who were diabetic, and recipients who were diabetic who were treated with the metformin at 3 time points, 4 weeks, 12 weeks, and 48 weeks. And they basically just measured lipid. And you can see that there's significant lipid accumulation that occurs pretty early, certainly by 12 weeks, in the hearts of individuals with diabetes, which is somewhat ameliorated in those who are treated with metformin. And shown here on the bottom is a Cox regression analysis showing that pretty much almost all the diabetic patients had clear evidence of lipid accumulation in their hearts. In addition to that, they also showed that there were toxic lipid intermediates, such as ceramides, that were also accumulating in the hearts of these humans, again, within 3 months and certainly definitely at a year post-cardiac transplantation. So the concept here then is that the healthy heart that's then put into this abnormal milieu that characterizes diabetes clearly shows evidence of mitochondrial dysregulation and lipid overload, which I think makes a very important contribution to priming the heart to increase the risk of failure at some point in the future. So what happens then to heart metabolism in diabetes? And this is data summarized from a lot of work that many others have done, both in animal models and also true in humans, that essentially in diabetes, the heart increases its dependence upon an oxidation of fatty acids. There's increased delivery of reducing equivalents to a mitochondria, which is impaired. And this then drives increased reactive oxygen species generation, which promotes mitochondrial uncoupling that drives up myocardial oxygen consumption rate. So the heart actually becomes less efficient. It actually does the same work at high levels of oxygen, so that if you then impose ischemia on top of that heart, you get much earlier injury and much more severe injury. So there are many mechanisms that are important that drive mitochondrial dysfunction in the context of diabetes. I've summarized these here on this slide. And what I'm going to focus on, so the list includes OXPHOS dysfunction, uncoupling, ROS production, altered proteomics remodeling, post-translational modifications like O-gluconation and altered acetylation. But I'm going to really focus on what we've learned about altered mitochondrial dynamics in this process. So what is mitochondrial dynamics? So as many of you know, mitochondria undergo repeated cycles of fusion and fission. And the fusion are mediated by a family of GTPases, such as OPA1, which drives inner mitochondrial membrane fusion, and mitofusins, MFN1 and MFN2, that drives outer mitochondrial membrane fusion. And in terms of mitochondrial fission, there are proteins that are responsible for that. An important one is DRP1, a protein that normally is cytosolic and then translocates to the mitochondria to interact with receptors such as FIZ1 and MFF and MID 4951 to then drive mitochondrial fission. So if one looks generally at multiple tissues in individuals with, whether animal models or humans with type 2 diabetes, what you tend to see is that there is a reduction in the expression of proteins that drive mitochondrial fusion and an increase in the activation of proteins that drive mitochondrial fission, such as phosphorylation of DRP1, as shown here, that drives fission. And this is associated with altered mitochondrial bioenergetics, oxidative stress, and insulin resistance. If one looks in cardiovascular tissues, such as the heart or in blood vessels, we see the same thing. A tendency towards signaling changes that drive mitochondrial fission, so reduced expression of these proteins that regulate fusion and increased activity of DRP1, usually mediated through specific post-translational modifications. So let me show you another picture from an animal model. This is a DB-DB mouse heart. On the left is wild type, on the right is a DB-DB heart. And what you can see very clearly is that the pattern of the mitochondria are also altered in these DB-DB hearts. You can see the crystal less dense. There's more like a cluster of grid pattern here. And of course, the orange arrows are pointing to lipid droplets, which are accumulating in these hearts. So we did a study that was published in 2018 that we really wanted to ask the question, what is the direct contribution of lipid overload to mitochondria in the heart? And to do this, we collaborated with Jean Schaefer when she was at WashU, and she had made a transgenic mouse that overexpressed the enzyme acyl-CoA synthase. And she had multiple lines in her initial report, one with very high level expression that developed a very rapid cardiomyopathy, but we wanted to look at the lower expressing line. And the reason why we chose that line was because when we measured palmitate biodistribution by PET scanning in these mice, we saw about a 2 to 3-fold increase in palmitate uptake, which is about what we would expect to see in diabetes. And importantly, we saw that there was accumulation of ceramide and accumulation of diacylglycerol. Very strikingly, when we looked at the mitochondria in these animals, we saw that there's a very dramatic remodeling of mitochondria. So if you compare the wild type to the ACS mice, you can see that really the appearance was of highly fragmented mitochondrial network. And so when we looked at that, we said, well, was this being driven by increased fission or decreased fusion or both? And so what we found mechanistically was that the overload of saturated fatty acids in this model led to increased generation of mitochondrial reactive oxygen species that did a number of things. First of all, it increased the proteolytic degradation of the mitochondrial fusion protein OPA1. And secondly, it led to the proteasomal degradation of a very important anchoring protein, ACAP121, that actually brings protein kinase A or PKA to the mitochondria to promote an inhibitory phosphorylation of DRP1, leading DRP1 to be constitutively mitochondrial, leading to mitochondrial fragmentation. So I want to now move and shift into a concept, which is as mitochondria become stressed. I think I've shown you examples in the diabetic heart that there's mitochondrial stress. I've showed you what happens with mitochondrial fission, that the mitochondria actually communicate this stress outward. And this is a busy slide from a review that was published by Martin Picard and Orion Shira. Orion is at UCLA, Martin is at Columbia, that really showed different kinds of signals that occur in the context of mitochondrial stress. What I'm going to focus on is this one that's talking about metabokines or mitokines, that as in the presence of mitochondrial stress, you get activation of an integrated stress response that leads to the transcriptional induction of molecules like growth differentiating factor 15 or GDF15 or fibroblast growth factor 21. So let me say a few words about each of those molecules. I'm going to show you some data that addresses this. So FGF21 ordinarily is made under physiologic circumstances in the liver, circulates as a hormone, and has multiple effects. Importantly, it has important effects on the brain to regulate food intake, appetite, alcohol consumption. It has important effects on the bone and the beta cells. It has important effects on liver metabolism. Importantly for this stock, it also has important effects on adipose tissue, specifically promoting the conversion of inducible white fat to brown or beige adipose tissue. Then GDF15 is also another stress hormone that is secreted. And has multiple pleiotropic effects in the cardiovascular system and also in metabolic organs. So, for example, in the liver, GGF-15 actually increases fatty acid oxidation, reduces fibrosis, reduces inflammation in the liver. In the beta cell, it inhibits beta cell apoptosis. In the vasculature, it reduces vascular inflammation. Um, in the heart, it has been shown to be cardioprotective, and it also reduces inflammation in white adipose tissue. When the levels get very high, as occur, for example, in preeclampsia, it actually can lead to severe vomiting, etc. And in cancer, when these levels can be very high, it can actually lead to wasting of muscle and white fat. So it's one of these kind of good cop, bad cop myokines, where that at lower levels they can have metabolic benefits. At higher levels, they can actually have significant adverse effects. So I'm telling you this because we landed into this almost by accident in a study that we had performed where we wanted to understand the role of mitochondrial dynamics in the pathophysiology of insulin resistance. And to do this, Renata Pereira, a colleague in the lab at the time, who is now has an independent lab, generated animals with essentially hypomorphic OPA1 allele in skeletal muscles selectively. So you can see here that she knocked down OPA1 expression by about 60%. You can see looking very closely at the mitochondria and skeletal muscle of these animals. You can see that relative to the normal mitochondria, we see this very nice lamellar looking cristae. In the knockout animals, you can see that you see this tubular cristae consistent with loss of OPA1. I should have mentioned earlier that in addition to mediating inner mitochondrial membrane fusion, OPA1 also plays a very important role in maintaining the morphology of mitochondrial cristae. So you can see that the cristae morphology are abnormal. What's really interesting in these animals is that when you put them on a high-fat diet, the skeletal muscle, well, the wild-type animals, of course, shown here on the top, became obese, but the mice with skeletal muscle OPA1 deficiency did not become obese. So they were resistant to diet-induced obesity, which is somewhat unexpected. When we measured the energy expenditure was increased as well. And also, these animals were resistant to diabetes and insulin resistance. So this is a glucose tolerance test, showing again with at the end of the high-fat feeding period, the high-fat fed mice become diabetic. You can see the increased glucose excursion shown here, whereas the knockout animals in the high-fat diet were protected from diabetes. Also, the wild-type mice on the high-fat diet became insulin resistant. You can see that by elevated levels of insulin, whereas the knockout animals on a high-fat diet did not become insulin resistant. So the bottom line was it turned out that their skeletal muscle was overproducing FGF21. And when we then knocked out FGF21 alongside OPA1, you could see that we reduced FGF21 levels in the muscle, but importantly, we also reduced the circulating levels of FGF21 as shown here in these animals. And you can now see that in these double knockout animals on a high-fat diet, FGF21 levels are no longer increased. But you can now see that the protection against obesity was completely lost. So they gained as much weight as the wild-type animals, and they were as diabetic and as hyperinsulinemic as the wild-type animals on the high-fat diet. Mechanistically, what we showed was happening was that there was the mitochondrial stress that was induced by reducing OPA1 actually led to activation of the integrated stress response, which, as you know, is a conserved response in every cell in response to various upstream mediators such as amino acid deprivation, infections, heme deprivation, and protein misfolding leads to activation of this stress response that ultimately leads to a series of phosphorylation events shown here, for example, EIF2 alpha, which ultimately induces an important transcription factor in the ATF family, shown here is ATF4, that then drives the transcription of a number of target genes. So we actually did an experiment where we inactivated ATF4 in the skeletal muscle of these OPA1 deficient animals. And what you can see is that, again, in the OPA1 deficient animals, there's increased FGF21, but when we knocked out ATF4, you can see that this increase in FGF21 was significantly abrogated. So the model then is that in the context of mitochondrial stress, and this is work done by Anata, who is an assistant professor at the University of Iowa, is that this mitochondrial dysfunction leads to this integrated stress response, induces ATF4, that in turn then it induces FGF21, and then FGF21 in turn has this effect systemically to lead to a resistance to diet-induced obesity and insulin resistance. So a few other things that we learned about this is that when you knock down OPA1 and in this case in skeletal muscle, we actually observed that there was increased mitochondrial ER contact sites. So if you just look here at some 3D reconstructions in control muscle versus OPA1 knockdown muscle, you're actually seeing, so ER is in blue, mitochondria in orange, that there's increased mitochondria ER communication as a manifestation of this mitochondrial stress. And this is the quantification shown here. And I'm not going to show you the data, but we also showed that ATF4 also drives the expression of proteins involved in mitochondrial ER communication in muscle cells. This is work done by A.J. Hinton. So the model then is that there is this integrated stress response. I mentioned ATF4, but also ATF6 XBP1 that then drives the induction of proteins involved in mitochondrial ER communication, in addition to increasing the levels of various myokines such as FGF21. So we then went on to look at this phenomenon in adipose tissue. And the reason for that is that there's adipose tissue depot, which is brown adipose tissue, which is present in abundance in rodents, but also present in humans. And what brown fat classically does is that in the presence of cold, there is activation of the sympathetic nervous system, increased lipolysis, increased fatty acids that then get metabolized in these mitochondria that have high levels of expression of uncoupling protein 1 or UCP1 that essentially leads to dissipation of the membrane potential and drives the generation of heat. And this may actually have an important relationship to cardiometabolic health, as was very nicely shown by Paul Cohen a number of years ago, where they looked at patients who had PET scans. So these are cancer patients who had PET scans, and they saw that there were some patients who had a lot of brown fat called BAT positive, and patients who have not so much brown fat called BAT negative. And then they looked in the medical record at what was the relationship between the presence of active brown fat or less active brown fat and certain cardiometabolic traits. And very interestingly, that they saw that in patients with less brown fat, they had more diabetes, more dyslipidemia, more cardiovascular disease, including coronary disease and heart failure and hypertension. And you can see this shown here in terms of the odds ratio relative to individuals with increased levels of brown adipose tissue. So what does mitochondrial dynamics have to do then with, brown adipose tissue activation? So Ori and Shirai at UCLA showed a few years ago in a very nice paper in EMBO that when you expose animals to the cold and you activate brown adipose tissue, there's mitochondrial fission that's actually required for the uncoupling and the thermogenesis. And he showed very nicely that this was mediated through increased phosphorylation of DRP1 that drove mitochondrial fission. So we decided to actually look in brown fat at what happened to OPA1. And interestingly, if you take animals and put them in the cold, there's increased expression of OPA1 mRNA, but reduced levels of OPA1 protein, and specifically reduction in the proportion of the long short of OPA1 to the short form of OPA1, because OPA1 undergoes this proteolytic cleavage. So it looked like another adaptation to the cold in brown fat was processing of OPA1. So Nata generated mice that were brown adipose tissue specific knockouts of OPA1. And what we observed in that paper was that we clearly impaired mitochondrial metabolism in brown fat. But interestingly, when we put these animals in the cold, they actually maintain their body temperature. And so you can see here in the gray bar that animals in the cold that were deficient for OPA1 actually maintain their core body temperature relative to the wild type animals in the black that actually dropped their body temperature by 2 degrees. And you can see here the core body temperatures were higher. So this was kind of a bit of a paradox because we said, well, we have impaired mitochondrial dynamics in brown fat, but yet, despite the impaired brown fat, these animals appeared to be thermogenic. And in fact, when we looked again in the circulation of these animals, we saw that there was increased FGF21 in the mRNA of the brown adipose tissue and increased FGF21 in the circulation. So I mentioned earlier that one of the things that FGF21 does is it leads to browning of inducible white adipose tissue. And so you can see here in these immunohistochemistry slides that the knockout animals had significantly more mitochondria in their inducible white adipose tissue. They had induction of uncoupling protein 1, induction of OPA1 in their inducible white adipose tissue. You see that the mitochondria just got much more beautiful looking, much nicer crispy, for example, consistent again with increased mitochondrial biogenesis. And there was also increased sympathetic input to the brown adipose tissue in these animals. And so that was the driver of the increase in thermogenesis that we observed. So this I just showed you just to remind you again that these animals maintain their body temperature. And then so we went ahead and we actually knocked out FGF21 in brown fat alongside OPA1. And now you can see that in these animals that no longer were we seeing this increase in oxygen, in systemic oxygen consumption. They were no longer able to, we didn't see this increase in core body temperature. And then in the cold, you could see that these double knockout mice now could no longer maintain their body temperature. So what this was really telling us then was that the mitochondrial stress that occurred in the brown adipose tissue induced this signaling response that increased FGF21 production, increased browning of inducible white fat that actually promoted thermogenesis. And when we got rid of FGF21, we lost that adaptive response. Now, what was really interesting is that all the data I showed you so far, summarized here, were in animals that were fasted. So you can see that in the fasted animals, so these are animals essentially that are double knockout for OPA1 and FGF21. They can't maintain their body temperature as shown here. Interestingly, if we did the same experiments in animals that were fed, right, then you can actually see that they actually were able to maintain their body temperature when they were left in the cold for a longer period of time. And so what this is telling us is that this FGF21 response is really a response to sort of increased bioenergetic stress in the brown adipose tissue of these animals, which is somewhat ameliorated when we do these experiments in animals that are constantly feeding. So what then has this taught us? So we learned, therefore, that FGF21 clearly is an important mitokine that is secreted in the face of mitochondrial stress induced by reducing OPA1, and that this clearly has important effects both to enable these animals to maintain thermogenesis and also to have systemic effects on systemic metabolic homeostasis and body weight. And of course, FGF21 is an output of this signaling pathway. So it wants to kind of confirm that ATF4, which is kind of core to the integrated stress response, at least at the level of driving the transcriptional changes. We want to see if this, in fact, was true in this pathway. So we generated mice that were lacking ATF4 and OPA1 in brown adipose tissue. So you can see now in these animals, we learned a couple of things. First of all, when we knocked out ATF4 in brown fat, we also saw that we did not induce UCP1 in inducible white fat. And then when we looked at another stress, which is high-fat feeding, we saw that the OPA1 knockout mice only, OPA1 only in brown fat, again, did not gain weight in a high-fat diet. You can see that reduced body fat mass, increased food intake. And you can see here that much, much higher energy expenditure relative to the control animals. But then when we knocked out FGF21 from these animals, we saw something really curious on a high-fat diet, that the animals still did not gain weight on a high-fat diet, suggesting that maybe something else may be, responsible for this phenomenon. And clearly, it's ATF4 dependent because when we knocked out ATF4, we lost this effect. So on the ATF4 upper one double knockout mice on the high-fat diet, they had diabetes shown in panel D. They gained the same amount of weight. They had the same amount of glucose intolerance. So there's something that ATF4 is driving that was defending these animals from high-fat diet-induced obesity, but it was not FGF21. So just to summarize what I've told you so far, then, is that we have this model where we knock out upper one in brown adipose tissue. We impair brown adipose tissue function. The response is that there's ATF4 mediated increase in FGF21 that certainly is responsible for the increase in core body temperature, resting metabolic rate, but not for the resistance to diet-induced obesity and insulin resistance. So the question then, what else could be happening? So we did RNA sequencing on the brown adipose tissue of these animals, and we saw quite a number of secreted proteins, but I want to focus you here on GDF15, which was also induced as well in these brown fat of these animals. And in fact, that was, of course, correlated with the increase in ER stress markers. And so when we looked at the circulating levels of GDF15, we also saw that in addition to so OPA1 knockouts increased serum GDF15. OPA1 FGF21 double knockouts had increased GDF15, but it was ATF4 dependent. So now we are now opening the window a little bit wider to a larger suite of cytokines that potentially could now be having a metabolic effect in these animals. And in fact, when we then in OPA1 as well as GDF15, again, what we observed was these animals were also not able to maintain their core body temperature as shown here, despite abundant UCP1 in their inducible white fat. And in fact, now when we looked at the growth curve response to a high-fat diet, you can now see that there was attenuation of the rescue. So again, this is wild-type mice. These are OPA1 knockout mice in the bottom, and now these double knockout mice are in between. So, which is saying that there's an element of the diet-induced, the resistance to diet-induced obesity that is being driven by GDF15, and also the rescue of diabetes that we saw in the OPA1 knockouts was now removed in the OPA1 GDF15 deficient animals as well. And interestingly, again, however, the animals still remain somewhat insulin resistant, saying that there's probably other factors that are also partially contributing to this phenotype. Now, importantly, again, just coming back to the critical role of ATF4, again, what we saw is that in animals that we put in the cold, you can see that ATF4 is induced by the cold, FGF21 is induced by the cold, GDF15 is induced by the cold. But when we knock out ATF4 and look at what happens, we actually prevent the increase in FGF21 and GDF15 as shown here. And then those brown fat ATF4 deficient animals, as ATF4 deficiency only, you can see that they absolutely cannot maintain their body temperature. As you can see, the body temperature analysis over time, when animals are left at 4 degrees, they don't increase FGF21 in the circulation, and they also don't have inducible white fat. So to summarize then, what I've shown you so far then is that ATF4 is a critical regulator of this response to mitochondrial stress. But then there are varying effects of either FGF21 or GDF15 in the regulation of insulin sensitivity, energy expenditure, glucose homeostasis, and resistance to diet-induced obesity. Another thing that came out in the RNA sequencing was a change in amino acid metabolism. And there's some nice work from Shingo Kajimura that has shown very nicely that brown fat amino acid metabolism is also important in thermogenesis. And what we actually saw is that in this ATF4 deficient brown adipose tissue, there's a broad repression of many of the proteins involved in amino acid metabolism in brown adipose tissue. So to summarize then, essentially what I've told you is that there's this dynamic regulation between cold stress, induction of the integrated stress response, requiring ATF4 that then leads to the release of certain mitokines or cytokines like FGF21 and GDF15 that have significant metabolic effects. So the question then was, okay, so we made a mouse that is missing OPA1 in brown fat. So that's interesting. But we said, what if we then put this together and inactivated OPA1 in brown fat and in white fat? And so just to remind you again, when we knock out OPA1 in brown fat only, we have these mice that have inducible white fat and they are thermogenic, they maintain their body temperature. But when we knock it out in brown and white fat, you can now see that these animals could not maintain their body temperature in the cold, as shown here with regards to their core body temperature. Despite, and you can again see that when you put animals in the cold, OPA1 goes up. But when you knock out OPA1 in brown and white fat, you completely lose the thermogenic response in inducible white fat, including the induction of UCP1. So it's showing that it's crosstalk between these pathways. And importantly, the white fat becomes lipodystrophic. So there's impaired lipid metabolism, impaired lipogenesis, shown here on the bottom, repression of many of the genes involved in lipogenesis. And in fact, there is also impaired lipolysis in these adipose tissues. There's less circuiting of free fatty acids. There is increased senescence in this adipose tissue, increased inflammation in this adipose tissue. So in a sense, what we've really generated here is a model that is somewhat lipodystrophic. And in fact, when we look at these animals at the level of whole body metabolism, we see that they get fatty liver disease and they have impaired glucose tolerance. So to summarize this part, then what we learned from these experiments then was that if you impair mitochondrial dynamics in brown fat and in white fat simultaneously, you have evidence of impaired mitochondrial function in both of those depots, characterized by decreased lipid synthesis and mobilization, increased inflammation and senescence, impaired mitochondrial function. And then this manifests itself as fatty liver disease, impaired glucose homeostasis, as well as impaired thermoregulation. So what I told you so far then is that the adaptation to alterations in mitochondrial dynamics is cell type or tissue relevant. And so the question then is, what happens then in humans? And so the nice thing about OPA1 is that there's actually a clinical disorder that is caused by OPA1 mutations called autosomal dominant optic atrophy. And so what Jen Streeter, who is an assistant professor at the University of Iowa, did was she went into a large clinical database called Trinetics, which essentially takes data from a lot of electronic medical records across the world, has over 250 million patients in it. And she made a query. She said, find me patients with this condition caused by a mutation in autosomal dominant OPA1, which is called autosomal dominant optic atrophy. And they had to be sequence or gene test verified. And then let's look at the cardiometabolic outcomes in this patients. And then essentially, the nice thing that you can do from this kind of analysis is that you can essentially pull an equivalent number of controls who are otherwise identical, except that they have wild type OPA1. So we then looked to validate that these patients, in fact, had the clinical disorders associated with OPA1 deficiency, such as hearing loss, visual disturbance, disorders of muscle and the CNS. And clearly, you can see that the OPA1 patients had the clinical manifestations that we would expect. So when we looked at cardiometabolic traits, what was really interesting is, and again, just to put this in perspective, these are about 25,000 patients with mutations in OPA1 compared to 25,000 controls. When we looked at body weight, the patients with OPA1 deficiency were less heavy, they're lower BMI, lower weight, lower BMI, but they were much more likely to have type 2 diabetes. You can see that shown here clinically across the age span. They're much more likely to be on diabetes medications. However, they were fairly well controlled in that hemoglobin A1Cs were normal. When we looked at hyperlipidemia, you can see they were much more likely to have hyperlipidemia. They were much more likely to be on lipid-lowering drugs, but they were fairly well controlled in terms of their LDL cholesterol, HDL cholesterol, and their triglyceride levels. Blood pressures were higher in the upper one patients compared to the non-affected patients, and they were much more likely to be on antihypertensive therapy compared to the non-affected patients. So what it's looking like, these patients have many cardiometabolic traits. Also, they had more fatty liver disease. So you can see that the patients with the upper one deficiency had a lot more fatty liver disease shown here in terms of non-alcoholic fatty liver disease. So we then want to ask about major adverse cardiovascular events. And before we do that, we wanted to look at one more thing because an important driver of major adverse cardiovascular events is smoking. And it turns out that these individuals who normally present as adolescents and are under medical supervision their entire life are much less likely to smoke relative to the control population. So that was encouraging to see because when we looked at major adverse cardiovascular events, in fact, we saw that the upper one patients had more ischemic heart disease. They were much more likely to be on antiplatelet medications, more stroke, much more likely to be on anticoagulant medications. And this risk was increased by up to threefold for ischemic heart disease, fourfold for heart failure, and twofold for stroke. So, and then also when we looked at heart failure, again, these patients had more heart failure relative to the non-affected controls. So this really told us in a very interesting way then that at a whole organism level, these humans with a primary defect in mitochondrial dynamics also leads to cardiometabolic disease. So what I hope to do in this talk is to kind of take on a journey, really showing you that as mitochondria become altered in metabolic disease, obesity, diabetes, insulin resistance, that there is potentially a direct relationship between, altered mitochondrial fusion, as shown here in these very selected patients who have a defect in mitochondrial fusion, i.e. OPA1 deficiency, and the propensity to developing cardiometabolic disease. And so with that, let me just acknowledge, after I show you these nice mitochondrial pictures, the lab. We moved to California. So when you go to California, for those who live there, everybody comes and visits us. It's my mother and my daughter, son-in-law, and son who came to help us unpack the lab in February, which is always nice. And then these are members of the lab here and members of the lab here who have contributed to various aspects of this work. And we have some important collaborators who have also contributed. And so, again, I want to thank you very much